We have a DNA sequence that looks like this. The top strand is the template strand and the bottom strand is the coding strand. After transcription, the mRNA sequence that results looks like this. The mRNA sequence looks identical to the coding strand with the exception that the T's are changed to U's. So this mRNA will travel to a ribosome where protein synthesis can occur during translation. Translation starts when the start codon is detected. The start codon is AUG and it codes for the amino acid methionine. We see that here is the codon AUG, so this will code for methionine. The next codon is GUC, which codes for valine, AGC codes for serine, AAG codes for lysine, and then we get to UAA, and this codon doesn't code for an amino acid, but instead is a stop codon. This indicates that translation for this sequence has ended and we ended up with a protein that has 4 amino acids. What would happen if a point mutation were to occur in the original strand of DNA? A point mutation is when a base is substituted for another base. You can have transition point mutations that are purine to purine and pyrimidine to pyrimidine, or you can have transversions which are purine to pyrimidine and pyrimidine to purine. Recall the purines are A and G while the pyrimidines are T and C. If there was a point mutation that resulted from a transversion where this C was changed to an A, you would get this mRNA strand. The resulting protein would look like this. As you can see, the protein is the same because GUC and GUA both code for the same amino acid, so no significant change is made. This is called a silent mutation. If there was a point mutation that resulted from a transition where this G became an A, the mRNA strand would look like this, and after translation, the protein would look like this. It looks mostly the same, but there is asparagine in place of serine. This is because the single base change made the codon AAC, which codes for asparagine. This is known as a missense mutation. If there was a point mutation that resulted from a transversion where this A becomes a T, the mRNA strand would look like this. The protein that results from translation would look like this. The protein only has 3 amino acids instead of 4. This is because the codon that was changed no longer codes for an amino acid and is instead a stop codon. This mutation causes translation to end prematurely and is known as a nonsense mutation. The last point mutation we are going to discuss is a read-through mutation. This mutation results when a stop codon is changed to a codon that codes for an amino acid. This would happen in our example if a transversion occurred in which the A was replaced by a T. This would result in this mRNA strand and a protein that looks like this. The stop codon UAA is changed to UUA, which codes for leucine. This protein would continue to grow until another stop codon is reached. This has been an overview of point mutations and how they can impact the protein that a sequence of DNA codes for.